Thanks, Arnie. Okay, so I'm going to talk about metabolism. How many of you guys know what this is? Glycolysis. Arnie knows. That's good. Okay, so this is the main pathway leading down from sugar. This cycle here in the center is the TCA cycle, so I'll talk mainly about those two things. And so obviously metabolism for mammals and all other non-photosynthetic organisms starts with what you eat. Plants and photosynthetic organisms can partly eat the sun. That's very special, and when they do that, they make molecules like this, which is sucrose, and this can be broken down into things like glucose and fructose. That happens in our GI tract before the food ever gets into the body proper. And then in the body proper, this is the glycolytic pathway. It takes glucose, which has 6 carbons, to pyruvate, which has 3 carbons, through 10 enzymatic steps. And from pyruvate, then in the mitochondria, which you heard about beautifully from Nav earlier, you can make acetyl-CoA, this key epigenetic metabolite that you heard about from Shelley. You also can make a waste product, lactate. And there's one important thing to learn immediately here, that if you count the atoms and the electrons, then glucose is equivalent to 2 molecules of lactate. Right? So these can be interconverted with the right machinery in a mass-balanced and electron-balanced manner, whereas pyruvate and acetyl-CoA are more oxidized. They have fewer electrons, and you have to put them somewhere. The place that you put them eventually is onto oxygen, of course, and this is why we breathe. And so acetyl-CoA enters the TCA cycle. This is a catalytic cycle that starts with a molecule with 4 carbons, and as it spins, it picks up the 2 carbons from acetyl-CoA and burns off 2 carbons. And this produces NADH, functionally high-energy electrons. And these electrons go through the electron transport chain in mitochondria that Nav talked about. And ultimately then this pumps protons. And when these protons re-enter the mitochondrial matrix, you get ATP production. OK, so that's the basics of metabolism. I'm going to talk about 3 things over the next roughly hour. I know it's getting late, and that dinner, real metabolism for you guys, is awaiting. OK. So I'm going to talk about concentrations of metabolites and fluxes of metabolites as 2 concepts. I'm going to talk about flux regulation and design principles in microbes, and then I'll talk about fluxes in mammals. So to measure metabolite concentrations, one of the first things my lab did was set up technology. And so this is one slide that goes through the basics of how you measure metabolite levels. Ultimately, the key instrument is a mass spectrometer. And what that does is it takes gas phase ions. So you have to have things in the gas phase, and they have to be charged, or ions. And it allows you then to separate them based on mass to charge by applying an electric field, and thereby get them separated and then quantitate the ions, ultimately using some sort of ion detector, which is a very sensitive measurement input. So the real work is in what you do both before and after the mass spectrometry step. And so ultimately, you start with something. If your life is easy, you may start with a biofluid like serum or urine. And I say that makes your life easy because those are more or less metabolically inert species. And so you can take your serum, and if you leave it on the bench for an hour, it's mainly metabolically going to be like what you started with. But if you're starting with cells in culture or a living tissue, those will change metabolically over seconds. And so you have to very rapidly stop metabolism. And so there are years of work in figuring out the way to stop things properly. You get samples that have your metabolites, typically in an organic solvent. And then before we ever do mass spectrometry, there are 2 things that we do. The first is to begin the separation process of metabolites using chromatography. Raise your hand if you know what chromatography is. A minority, OK? So chromatography is a process where you put some chemicals at the top of a column, and then you flush liquid over the column, flushing the whole time, and there are beads in the column. And the things that stick better to the beads stay in the column longer. The things that stick less well to the beads come out faster, okay? And so that's the basic process of chromatography, and when it works well, you get a very orderly output. So nice sharp peaks like this, one after another, marching off of the column. If you do this with all the metabolites in a living organism, you get something that looks like this and this and this and this, and it's all a big mess, and that's why you need the mass spectrometry also to figure out what's going on. The problem is that if you do only the mass spectrometry, you also get a mess. So it's these two orthogonal dimensions of separation that work hand in hand. The output of the chromatography column are little droplets of liquid, and these need to be converted into gas phase ions. There's a Nobel Prize for this, something called electrospray ionization. You apply a very strong voltage to the output of the column, and that very strong voltage produces tiny droplets that are charged, and as they evaporate, you have more and more charge per unit volume, and eventually those droplets then explode due to like charge repulsion, and that gives you your gas phase ions. Okay. Then you measure them as a function of both time and mass coming out, and ultimately we have to associate these peak signals with particular metabolites and their concentrations. And so we started doing this with some very simple experiments where we took nitrogen away from bacteria. And there was a lot of literature on what happens when you take nitrogen away from bacteria, and it said there really should be one metabolic hallmark, and that's an amino acid, glutamine, should go down. And we did this experiment many times, and we got very, very consistently the result that glutamine was going up. Okay? And so this is a classic situation in biology. Okay? And so eventually we were able to figure out the difference between our experiments that showed glutamine going up and the true experiments, it turns out, that showed glutamine going down was how quickly we were stopping metabolism. So here we thought we can spin our bacteria to the bottom of a tube and then make the metabolic measurements. The process of spinning to the bottom of the tube, very importantly, didn't make things erratic. It made things very consistently the opposite of the way things really are in growing bacteria. And so that's an important message for you guys to have in your mind when you think about biology experiments. And so after we figured out a quicker way to stop things, we could get the correct answer. And ultimately, we're able to advance this technology to be able to measure many things. And one that you heard about that ties in nicely with the talk today that I'll go through quickly is what are the metabolic products made by weird enzymes that are mutant in cancer. And you heard from Shelley and from Nav also about this mutant isocitrate dehydrogenase. And so ultimately, this is the kind of data we got when we looked at signal from cells expressing the mutant isocitrate dehydrogenase versus the wild type. So we get many points along the diagonal. Those are all different metabolites that don't change, and we get exactly 3 points off of the diagonal. And it turns out all 3 of those points come from a single metabolite. And that metabolite is this metabolite here, 2-hydroxyglutarate. It's 2-electron reduction of a normal metabolite in the TCA cycle called alpha-ketoglutarate. So that's peak A here. There's a more abundant peak B, and that's where water is lost from this in the process of forming the gas phase ion. And then there's a less abundant variant C, and that's where one of these protons is replaced with sodium. So ultimately, knowing that this was the metabolic error product, people realized that they could try to turn off this mutant enzyme because it was making this oncometabolite or cancer-causing metabolite. And this is the drug that looks nothing like alpha-ketoglutarate or 2-hydroxyglutarate that was eventually made by this company, Agios, and FDA approved, and is helping people with AML.
Can I ask a question?
Yes.
Go back 2 slides. Are those forms B and C in vivo forms or part of the?
These are purely analytical artifacts of doing mass spectrometry. So they're formed during the ionization process.
I just wondered if they had different properties.
No, they're just analytical fakes. Good question. Okay, so now I'm going to talk about fluxes. So why care about fluxes? Well, fluxes are what actually make metabolites. One of the reasons metabolism is interesting is there's only really 2 good forms of bioengineering. One is immune system engineering, where you can make things like CAR T cells, which you may or may not have heard about yet. And the other is metabolic engineering, where you can make things like ethanol and many other valuable products. And this is purely a flux control problem. You have an input and you want to get the desired output. The biggest diseases involve flux control errors. Okay? So diabetes is fundamentally a disease of excess glucose production, and cancer is fundamentally a disease of too much anabolism, building too much stuff. And so, but there may be other things in addition to the substrate that feeds into the reaction that matter. But if we measured this rate of reaction 1 under many conditions, sub 1, sub 2, sub 3, we can see if those, for example, align in some simple way, like the velocity of reaction 1 as a function of A follows a Michaelis-Menten type curve. And if we make these measurements of fluxes, these reaction rates and concentrations for many reactions at once, this is an efficient way to learn about how the whole system is organized. And so, in general, this relates to a broader hypothesis that I'll present to you guys to think about, which is that when we think about putting together omics scale data, big scale biological data, with the goal of learning about biochemistry or the physical basis of life, I think it's most effective when we look at closely related levels of omics data. So for me, after years of trying to do this, it's very hard to relate transcripts, for example, and their levels to the functions of metabolism, because there are too many intervening events. A transcript may or may not get translated. The translated protein may or may not get degraded. If the protein is there, it may or not be phosphorylated, and so on. But if we start with things that are closer together, like enzymes and metabolites, we do think that these two things control fluxes, okay? And so at least then we can test these hypotheses in these very, very complicated and messy biological systems in a sensible way, where disagreement, rather than just being something we throw our hands up at, can point to new regulatory principles, for example. And so this is the one that we've worked on. Can we express flux as a constant function of enzyme and metabolite levels? And we've done this across 25 different steady states in yeast. Hopefully it's going to go forward. And so for each reaction, these are what I was calling V's on the board. Okay, there's a rate, and it depends on some constant, which is the catalytic constant of the enzyme, the concentration of the enzyme. This is something we've measured. And then some Michaelis-Menten type formulation here. I know this is a little bit more complicated than what I wrote on the board. This minus part here reflects reversibility. All these things are thermodynamically reversible. And then both substrate and product can almost always compete for the active site of the enzyme.
So did you measure the absolute values of concentration or relative values? This is the readout that the machine itself speaks, not the concentration. So what did you use to do this equation?
Right. So you can do all this in arbitrary units if you want, but we also can convert those peaks into absolute values by spiking in standards. And so we know the absolute values for all the metabolite concentrations we're talking about here.
You don't have to. So if you work with this equation, you can use arbitrary units.
Yeah, this equation works fine in arbitrary units. It just means that these k values are going to be in arbitrary units. Yeah. Good question. Okay, and so then we have these kinetic parameters, but because we have for each of these 25 different fluxes measured with different enzyme and substrate and product levels, we have a lot of data points relative to this relative few number of free parameters. We can optimize the free parameters, and then we can see how things work. And so for some reactions, we see a very nice agreement between the measured fluxes, which are shown here in red, and the output when we fit these parameters and say, what do the enzyme and metabolite levels tell us? So everything is copacetic. And then for some other reactions, we see a big disagreement. So here we know the rates of these reactions are changing a lot over the conditions, but we couldn't find any parameter sets that would input the enzyme and substrate and product concentrations and produce the proper changes that we experimentally observe in the output. So that means our function is missing an input variable or missing a Hill coefficient or something like this. Questions? Okay.
Whether if the case doesn't work is because this dynamic that you're presenting there is not a good one. I mean, this strategy, let's say, of fixing the fluxes because you said you're missing some coefficient. It could be because the dynamics. The formula is correct.
It could be because we've mismeasured the fluxes, you're saying.
Yeah.
This is all done at a...
Because you need some other mechanism on top of the flux. You keep repeating that fluxes, and then you need something else. That's the reason why it's that.
I guess I don't fully understand. I mean, fundamentally, the metabolite is being made by some enzymes and consumed by some enzymes, right? And so ultimately, the flow through those enzymes has to be controlled by something. Now, it could be the metabolite levels, it could be the level of the enzyme, it could be modification of the enzyme, but I think the...
Either back reaction effects from the concentration of some metabolites or whatever that make things a little bit more entangled.
Right. So in that case, I would say that would be a category I'd say that we've mismeasured the flux. It's some assumption we've made, and that's what I would call it personally.
How do you deal with changes in enzymatic activity, for example, post-translational modifications or changes?
Right.
How do you put that in the system?
Yeah, this is a great question. And so right now I'm being totally blind to that. And so blind to that, some things work fine, other things don't. And so now what we did is we said, let's look at the concentrations of all the other measured metabolites and see if by introducing one of them as an allosteric effector, can we take the cases that look like this and make them look like this, all right? And so we did that computationally, and we found that for this enzyme that I was just talking about, which sits right at the bottom of glycolysis, we needed an allosteric activator, in this case fructose bisphosphate, and this was known very well from 50 years of literature to be an allosteric activator. But the same analysis also predicted that there's an allosteric inhibitor of this enzyme, which is a TCA metabolite citrate, which had actually not been proposed over the 50 years of studying the enzyme to matter. And so, yes?
Wondering about a bit, like, what modeling went into those predictions then? And was it a sort of try a new model, see how it fits with new stuff, or do you have a more general detecting what effect could be present in the network?
So it's a very good question. The simplest way to think about it that's reasonably accurate is to say that we're just going to add a scaling parameter, again in a saturable way, where either you have a saturable activator or a saturable inhibitor. That's another metabolite. And so we have the measured data on all those metabolites, and so we just multiply here by a saturable function that's either an activating function or inhibiting function. And we test that out. And we see, is that statistically significantly improve things or not? All right. Okay. Yeah.
Is this system thermodynamically closed or open?
Thermodynamically closed or open. So this is you mean, is there energy leaking from the system? Yeah, this system is leaking energy. So these are, yeah, these are yeast sitting in a chemostat where we're constantly pumping in fresh nutrients. And then the yeast are spitting out waste, and the waste is running over the top of the dish and being thrown away. And the whole thing is leaking heat to the outside world.
So by introducing more variables, I mean the concentrations of other metabolites, you try to make the prediction data better. So my question is, I guess there might be some underdetermination problem. I mean, maybe you find this, metabolite A and B can correct the curve, equally well. How do you resolve that issue?
Yeah, so we had a lot of problems like that the different metabolites are kind of correlated across conditions.
Yeah, for example, you measure the fluxes that level off, and you find some other metabolite level off, and you introduce that metabolite as the
So I think the cheating way that we did it here, and we did cheat a little bit, is we took candidates. And so we took the candidate, any metabolite that had been suggested to regulate that reaction in any organism. So we did kind of a Bayesian integration with prior knowledge. I think the better way to do it is, first of all, to introduce mutants and make more measurements, and then you'll get more linearly independent inputs, okay? And so that would help. And then also you can measure on the back end, if you have 4 candidates that are kind of about equally good, you can do this experiment where you just test them, okay? You express the enzyme and you put in your regulator. And so here you see, this is what was well known. As you add more fructose bisphosphate, the reaction goes faster, but it doesn't matter how much you have, citrate will always turn off the enzyme. And yes, we have to do 5 or so of these experiments to find one winner, okay? So 4 out of 5 predictions are bad, but that's not too expensive for us. And so ultimately, I think this is a nice expression of how this system works. People had studied carbon limitation a lot before, and so the little dots are when the cells are going slowly because they have very little carbon, and as the dots get bigger, the fructose bisphosphate gets bigger and the reaction becomes faster. But in nitrogen limitation, when you start with very little nitrogen, you still have lots of fructose bisphosphate. The reaction is slow, however, because you have a lot of citrate, and as you put in more and more nitrogen, the reaction goes faster and faster because the citrate levels fall. So depending on which environmental pressure you're facing, you have a very simple way of controlling. It doesn't involve any complicated integration or multiplication. It just involves a dominant control variable depending on the environment. And so ultimately, then, we're able to think more generally about what controls metabolic activity in actual cells across physiological conditions. And that really depends on 2 things: the sensitivity of the reaction to the input. So when we have this Michaelis-Menten type of equation here, so we can derivatize this with respect to E, right? And we always get a factor of 1. So there's always a kind of unit value input there. But if we're sitting in where the concentration of A, for example, is very big, then this sensitivity to A may be very low. Okay? And people had thought about this a lot in terms of metabolism. Okay? But what they'd failed to think about was the fact that the real control variable also has to show biological variation. Okay? It doesn't matter that the enzyme, hypothetically, in the Michaelis-Menten equation, has a control coefficient of 1, right? If the enzyme level isn't varying. And so here we have a simple multiplication to integrate those factors, the sensitivity and the biological variation. And what you see is that when we then plot here the impact of reactions that are controlled by enzyme levels, controlled by allostery, and controlled by substrates and products, most reactions are controlled mainly by substrate and product levels. Everything works very simply, kind of by mass action. Right? This is why these systems can exist, because they work fine based on mass action, and so they don't need regulation. For the key committed steps, there tends to be regulation, allostery that matters, enzyme levels begin to matter more, but these are really the special reactions. And so ultimately, I continue to view metabolism mainly as a self-regulating network, right, that's controlled by the nutrient environment, for us, controlled by what we eat, right, and then it's fine-tuned by all the wonderful things that you heard about from Shelley that control ultimately transcription and the levels of enzymes. All right, so this is where we got in yeast. And so here are some questions for you to think about. So can we build more robustly predictive steady-state models? So we have these flux balance analysis models, but can we make them more robustly predictive? They're pretty predictive for E. coli under carbon limitation, but that's not that great because most metabolic engineering is done in yeast that behaves a totally different way. can we build dynamic models? And if we build dynamic models, what would the value be? And that's something I wrestle with a lot, because I'm pretty sure the answer to this is, of course, we can build them. We can probably even build ones that make accurate predictions. But what are we really learning, especially as we build bigger and bigger differential equation models? Understanding the output of the models may not be so much simpler than understanding the output of the organism that we can also measure. Okay, so now I want to talk about us as we get towards dinner. So I'm going to talk about the very basics. Yeah.
You do this sort of analysis for glycolysis.
Mm-hmm.
Based on your work, do you have a consensus as to, I think there's been a long debate about the 10 steps, which of those steps do these control coefficients,
Mm-hmm.
control of the flux, right? There's a ranking order of the 10 steps. Some are neutral, some are very little, some are predicted, like the first step, or gap pH being
Right.
the largest control. Does that pan out?
What I mean, you may or may not know, we did a very systematic analysis of this recently in mammalian cells, and we at least got what I would say is a clean answer. And that is that taking glucose into the cell is very important. Phosphorylating glucose the first time is somewhat important. Doing the second phosphorylation step, which has been classically in textbooks called the key regulated step in glycolysis to make fructose bisphosphate, is very important. And then ultimately spitting out lactate through the MCT transporter is important. And everything in between, except for those 4 steps, did not seem to matter in our analysis.
At each of the steps going down glycolysis, there are drains that go out
Yeah.
to other areas which are not being incorporating in.
Yeah, I think one way to think about that is those drains are like the effect of your house on the water main. They're very, very small. And so there's been a lot of talking about those drains. And in certain microbes, some drains are big. There's something called the pentose phosphate pathway. There are microbes where that takes 60%, and it really, really matters. But in yeast, it's not taking more than 4 or 5%. In mammalian cells, we never see it take more than 1 or 2%. So,
Neither any LDH for energy regeneration or consumption by gap pH.
gap pH. So the key thing about this analysis of sensitivity to enzymes is that if you pour on more and more enzyme, and the step is not limiting at all to begin with, you'll see a sensitivity of 0. But if you take that same enzyme and start taking its activity down and down and down, okay, then it will become the bottleneck enzyme. Okay? And so I'm totally open to the possibility that gap pH is the bottleneck enzyme in oxidative stress. We didn't study oxidative stress. Okay? In normal conditions where there's not oxidative stress, we don't see it being the bottleneck enzyme. Doesn't mean there's not some cell type where it is. Okay, so we're going to talk about the basics of mammalian metabolism and tell a few half jokes before we get into a little bit more math. So this is the town that I spent my 16-year-old year in, and I spent it there with my parents in a small apartment with my substantially younger brother, and I felt a little cooped up as a 16-year-old with their parents can. And so for me, freedom was riding on the bus, okay? And I was from a small town in America, right? And all of a sudden I was in Scandinavia on the bus, and I was 16 years old, and so you can imagine I had a lot of things that attracted my visual attention on the bus, okay? But among these was a sign, and this was a public health message that said, Spis mer brød, okay, which stands for eat more bread, okay? And so that was a public health message, okay, in the 1980s in Norway, all right? That was the thing to do. And so I wanted to show you the actual sign, so I typed this into Google image search, and Norwegian signs from the 1980s are not well represented there. But I did get a top hit, and the top hit is a modern Norwegian book, right? And this book's title is Bedre uten brød. That says better without bread, okay? Live long and healthy with a low carbohydrate diet, okay? And so this is the extent of our knowledge of what to eat. And so this is my grand challenge to you guys. Use mathematics, physics, physical constraints to help us all figure out a little bit better public health messaging so people are not making these same jokes 30 years from now. And so that's my challenge statement. I can put it in a little bit less silly of a format. And so a cure for cancer would, of course, be the most transformative thing in the world for people who are fighting the disease, especially younger people who are fighting the disease. On average, it would be worth about 5 years, between 4 and 5 years of life expectancy in the US. Okay, that's a huge deal, right? But it's also not that big when you think that there are studies, there's a very nice study pretty recently from JAMA, that compare lifestyle factors matched for socioeconomic status and show very clearly that lifestyle factors, and diet is almost certainly one of the biggest ones, diet and exercise, which are both metabolic factors, are ready today to produce those kind of gains. And that's without any smart diet or smart ways of integrating diet and exercise or personalized diet. And it just shows you how important these questions of lifestyle and what we eat are. Okay, so let's look at what mammalian cells eat. We can do the kind of analysis that I talked to you about where we pour on a bunch of different isotope tracers. Those are our food coloring equivalents in the river examples. Measure what comes in and comes out. Put this all under a flux balanced framework and tell you what fluxes are. So here the big arrows are the big fluxes, and so the biggest flux in the mammalian cells we're measuring here is from glucose to excretion of lactate. The biggest flux into the TCA cycle is from the amino acid glutamine that I mentioned in the beginning as our thing that goes up and down with nitrogen in E. coli. And then you can see how these change. So for example, if you express the most important, probably oncogene, then you get increased flux from glucose to lactate and decreased glucose entry into the TCA cycle. We can measure all of this, it's good. And we've measured this now across many different mammalian cell types. Some of them transformed, some of them untransformed. And the problem was, as we did more and more cell types, we kept getting the same map. Right? The biggest flux is from glucose to lactate excretion. The biggest contributor to TCA is glutamine. And so you say you get the same answer for 50 different mammalian cell types. I guess that's how mammals are, right? Except for this very big problem. Right? None of us, when we go to the bathroom, pee out massive amounts of lactate, okay? So fundamentally, we burn all the carbon that we eat, or we store it as fat, okay, hopefully in moderation. And so fundamentally, the metabolism of our bodies is totally different than the metabolism of our body cells in a culture dish. Put another way, we're seeing a majority of the energy from the TCA cycle in cultured cells being made from glutamine, but in reality, in our diet, glutamine is about 1% of caloric intake, okay? So 1% of caloric intake cannot be our main source of TCA energy. And so ultimately, we've been trying to reconceptualize how metabolism works in mammals. And I think for this audience, I'm going to go through these equations at least hopefully well enough for some of you to understand them. So the big master equation is that whatever we eat and breathe in has to balance fundamentally with what we pee out and breathe out, okay? So the metabolic activity of our body has to turn this stuff into this stuff. That's the big control equation. Then each tissue of the body has to be under flux balance. And there are two parts to that. This is what we call stoichiometric matrix, that matrix that's mainly zeros and ones and minus ones that says whether each metabolic reaction is making or consuming a metabolite. And that's multiplied by the vector of fluxes that occur in that tissue. Okay? These together as a product tell you the metabolic activity of the tissue. And that has to be balanced except for whatever that tissue is spitting out or taking in. Right? And that's this F vector that shows what the tissue is taking from the circulation or giving back to the circulation. Okay, so this is a zero vector. This thing is the vector of the metabolic activity of every reaction in that tissue. And then this vector is what's coming in and out. And then, of course, what comes in and out of different tissues in the body has to balance except for what you're eating and what you're excreting. And so those are the sets of balance equations that form the fundamental physical context for us thinking about these problems. Now, all of this would be great if we just said, now we have a very lovely metabolic objective, like we want to grow as fast as possible, the way E. coli does, okay? And then we could optimize everything. Except, of course, that's not how our bodies work. So now we have tons of unknown fluxes in these equations, and we have no simple organizing principle like optimal growth to say what the actual fluxes are. And so we need measurements. And so one type of measurement that's very useful is a measurement of what's the difference of metabolite concentrations between the blood coming into an organ and the blood coming out of an organ. And we've made those measurements now systematically for all easily detectable metabolites in pig, and we're just publishing that. But to really figure out what happens inside the organs, we need isotope tracers. And so we introduce these isotope tracers into mice that are catheterized so that we can drip, drip, drip in tracer, so non-perturbative amounts of different nutrients with extra neutrons, so carbon-13 or nitrogen-15, so we can see them by mass spec, okay, into their circulatory system. And so one of the things we do by dripping in these isotope-labeled nutrients is measure something that I call circulatory turnover flux. Okay? And this is a measure of total metabolite production by all tissues of the body. And you can think of it this way, that tissues in the body are making and consuming metabolites. Right? And ultimately, these two processes have to be balanced. The total production by tissues has to balance with the total consumption by tissues. And we can measure this by dripping in a tracer amount of the labeled metabolite and then looking at how much this tracer is diluted by the endogenous production. So the labeling that we get out reflects the rate you're dripping in the tracer divided by the rate you're dripping in the tracer plus the endogenous production rate of the metabolite. And we can just invert this equation, of course, at pseudo-steady state, which we reach metabolically over about 100 minutes, to get this circulatory turnover flux. Okay. So we've measured this now for every major circulating nutrient in mice, which is, for all of Arnie's joking, about the same as humans. Okay? And this gives you a perspective. So this is what the mice are eating. They're mainly eating carbohydrate. They're also eating some protein, amino acids, and they're eating a teeny bit of fat. And this is what's flowing around in their circulation. And we'll focus on the fasted state, maybe it's most interesting. So there's some glucose floating around that corresponds to the carbs. It's more when you're fed and less when you're fasted. That makes sense. There's a big chunk of something that's related to glucose, lactate, that's flowing in and out of the organs of the body, even though you're not eating lactate. There's some amino acids, a little bit more flow than you would expect based on the amount in the diet, but nothing too crazy. And then there's a walloping amount of fat circulating in the body, even though you're eating very, very little fat. And so ultimately, this reflects two very important, what I would call futile cycles, or at least semi-futile cycles, metabolic cycles. One is where in your body you have stores of fat, triglyceride, that get broken down into free fatty acids, the things in yellow here, plus glycerol, which is in black here. You're not mainly eating these, you're mainly cycling them in and out of the fat stores of your body, and that produces this big flux in the bloodstream despite the small dietary intake. The other cycle is glucose is being converted into lactate in the circulation, which is converted back to glucose around and around. And that's why you have this big red component here, even though you're not eating lactate. And so probably the most shocking part of this to people who'd studied metabolism a long time is there was a very clear view in the textbooks of what happens to the food you eat, especially carbs. You eat carbs, they're made of glucose. When cells need energy, they take in the glucose, they turn it to pyruvate. In the TCA cycle, they make CO2. You put CO2 back in the blood, and you breathe it out. Right? So in this picture, there is no role for circulating lactate. Right? But here you see the circulating lactate fluxes are on the same order as the glucose flux. And so fundamentally, this says that circulating lactate is an intermediate in the catabolism of glucose. Right? And so rather than cells eating glucose and burning it to CO2, some cells eat glucose, turn it into circulating lactate. They're specialists in glycolysis. And the rest of the body, when they need energy from carbohydrate, takes in circulating lactate and burns it to CO2. So this is a conceptual idea, and it leads to a very clear prediction. And the prediction is that if we start dripping lactate into the mouse blood, then we ought to see that lactate label the TCA cycle in organs a lot, if that's what's actually being burned, okay? And so this is an experiment testing that. And so we drip in the lactate, and we call the lactate labeling in the bloodstream 1, because it's what we're dripping in. Then you look in the TCA cycle, and in every organ of the body, you see there's a lot of TCA labeling from the lactate that you're dripping in. What if instead you drip in glucose? Well, if you drip in glucose in the brain, you get a lot more labeling from glucose than from lactate. The brain loves glucose. Right? This is one of the major known facts of mammalian metabolism. Every other organ seems to also use glucose, but less than it uses lactate. Right? So normally in biology, when you see an effect like this is a time and a half as big as this. You say, time and a half, okay, that's not an important effect in biology. But here, I'll give you an argument for why this 1.5-fold effect may really matter. So we said, what if all of this labeling here is kind of a fake? None of these organs actually take carbon from glucose directly into their TCA cycle. What if the only reason we're seeing labeling here is when we drip in the glucose, we're getting lactate labeled in the bloodstream, which we measure here, okay? And so I'll show this for the case of the liver to keep things simpler. So when we drip in the lactate, we call the lactate labeling in the serum 100%. The liver labeling that we measure in the TCA cycle is 30%. So now we're going to drip in glucose instead. It produces lactate labeling in the circulation that's 60%. And if the only way to get into the TCA cycle here was via the lactate in the serum, then we can predict that the liver TCA labeling should be 60% times 30%, or 18%, which is exactly what we see. You guys with me? All right. And so many of you probably already have in your minds that this could be scaled. Okay? And so this can be scaled where we can more systematically say when we drip in labeled glucose, it might make circulating lactate or circulating glutamine or circulating other things. And then we can write down equations that are weighted. So we call the circulating glucose labeling from glucose 1. The lactate labeling we can measure is 0.63. The glutamine labeling is 0.11, and so on. And the sum of these things has to give us the ultimate TCA labeling that we measure. Right? So this is the direct route, so to speak, and these are various indirect routes.
Can I ask a question?
Yep.
Why is it, given this, why is it when we do a glucose tolerance test, we're measuring glucose? Why aren't we measuring lactate? I mean,
It's a very interesting question, right? So
I mean, the medicine clearly has its way, but this says that measuring glucose is not necessarily the thing you want to know.
it's certainly not the be-all and end-all. I mean, part of a glucose tolerance test is the ability to store glucose as glycogen, which really is glucose metabolism. It's not glucose burning. Okay, so that's part of the answer. I don't know. I believe that insulin has 3 major actions, basically, to allow glucose to enter cells and be stored as glycogen, to suppress fat release from adipose, and to allow tissues to burn lactate. And so no one ever thinks about insulin as a master regulator of lactate metabolism, but we're going to try to show that. But glucose and lactate go hand in hand. They're just partners in crime. You know, one makes the other very, very fast. And so for you guys, I'm sure you saw this is not a very good equation because it has these 3 direct contributions as unknowns, right? And only one output measurement, this TCA labeling. But you can imagine if we do the same experiment dripping in glutamine and lactate, then we get a 3x3 matrix, as shown here. This is beautiful work by Tony Hui, who's soon starting his own lab at Harvard. And ultimately, he has made this into an n by n matrix where we can look at the direct contribution of every circulating nutrient to every tissue's TCA and thus energy metabolism. And this is kind of your master map. I won't belabor it because you're ready to fill your own TCA cycle soon. You see here in brain, however, the large glucose contribution. You'll notice that in the fasted state, which is the second bar, there's no other organ in the body in which there's a direct glucose contribution to TCA. You'll see that muscle in general loves eating fat and hates eating amino acids, whereas many of the visceral organs, like things in your belly, actually are good glutamine and amino acid eaters. Okay, and another thing you'll notice is that every major circulating nutrient here, except for this one glycerol, ends up being burnt. Glycerol, I said, is coming in and out of the bloodstream very fast so that you can constantly package fat into triglyceride and release fat from triglyceride. And so the one you never burn is glycerol, because the only time you dump it out is in the fasted state. And in that state, the last thing you want to do is burn glucose units. You want to return those to make glucose, because ultimately that glycerol to glucose is what feeds your brain in a prolonged fast. Okay, so I said those things. And so here are key questions for you guys. Why does the brain love glucose so much? And why doesn't the rest of the body? And I'll just quickly comment that I think part of the reason why the rest of the body has been wired to not use glucose in the fed state is for conservation. Part of it is so that you can regulate glucose metabolism for higher purposes. We know if glucose goes too high, you get a lot of damage. We know that you need glucose to do anabolism. So when you need to grow, if you need to fight an infection, you need glucose. If you need to fight redox stress, like Nav talked about earlier, you need glucose. And so these are the special things that glucose allows. Making energy is boring, okay? And that's the function of lactate. The other thing that happens with these cycles that we're learning, and maybe use some physicist help on, is that we think by doing all these seemingly futile cycles, what you do is poise metabolism in a state where you can go have a very different meal tonight, and your internal metabolism hardly has to change. You can go for a run, and even during the run, your lactate metabolism in the bloodstream doesn't have to change much, because it's so basally revved up. It's always prepared for you to go for a run, a walk, eat a healthy meal, eat an unhealthy meal. And so ultimately this is achieved through just exchange reactions. So ultimately, we're not making or breaking anything here. What we're doing is very rapidly trading something in the cells, pyruvate, the product of glycolysis, with lactate in the circulation. And this helps everything, because now we don't have what's been a chronic problem in metabolism, that when glucose makes pyruvate, you have extra electrons that you have to deal with. You just make lactate, trade it with the circulation, and when you're ready to deal with those electrons, you can burn the lactate into the TCA. And so this makes glucose metabolism tunable, controllable for higher purposes, so to speak, and energy metabolism simple, robust, and the same across many conditions.
I've always learned that lactate builds up in my muscles when I use them, right? And that's bad because, right? That's what's happening.
Well, what I can say, and we're in the middle of this, so I don't want to overpromise, okay? But what we see is that if there's no doubt that there are experiments that show if you really push muscles to failure, it's associated with lactate buildup, right? But you can have mice go from being totally lazy to running at a good rate, not far from their maximal rate for 15 minutes, and we don't see big changes in whole body or muscle lactate levels or fluxes. So the flux is big enough in the basal state that it just absorbs the added lactate that's made from the expenditure of running. And so that was a result that we just hated for 2 years. I just could not get my head around it. And now I'm like, I think this is the design principle, actually.
Go back to that, one of the final figures you had there. Yeah, this one. I was wondering just about how you collect the data. Are you looking at many mice and then averaging over them? Did you take a single mice and look at all the different organs at the same time?
For each mouse, we take all the organs, but to do this, to fill out this matrix equation, we have to infuse all these different things And we can't infuse all these different things at the same time because we only have carbon 13 as our reporter at the end of the day. So we have terminal experiments with each of these infusions looking at like 6 mice per infused nutrient, something like that. Yes.
So, and I know that there are buffers for like a big change in lactate production and consumption will affect pH really rapidly.
Mm-hmm. Yeah, I mean, there's definitely a big system for pH handling, lots of things that pump protons in order to keep that under control. And so, but this may be another advantage of having a minority of cells do glycolysis, because if you're not doing glycolysis, you're not subject to that stress of turning something that's pH non-reactive into an acid, right? Which is a challenge that glycolytic cells have to face. And we're trying to figure out what the glycolytic cells in the body are right now, but that may be a lead, actually. That's an interesting point you made.
It's a very nice strategy to decouple what we eat from how we spend it. Do you think this is purely mammalian?
I think it goes pretty far back. You know, I keep toying with the idea of doing some experiments to show that yeast also act this way. This is kind of my, it kind of goes with the basic behavior of yeast. When you have glucose, you turn it to ethanol. Then you eat the ethanol. And so I think the entire time they're bringing carbon into the TCA, but my guess is that carbon is really environmental ethanol carbon, even during the time they're eating glucose. And so I think this goes all the way back to the beginning of eukaryotes, basically.
Yeah, so just back, I know you explained briefly, but just back to the question of why the brain is the one that's, you know, eating up all the glucose. And like, so I understand the advantage to having the lactate, you know, cycling around, and it's kind of like when you talk about energy consumption and like the grid and stuff, it's like having that constant energy source that's not dependent on income. But why exactly, again, what would be the disadvantage to the brain using the lactate, I guess? Or is it just to always have the glucose there?
You know, again, I have a bias, and it's not a well-proven one, but my bias is that when you fire a neuron, you need to repolarize quickly. That requires a lot of energy for sodium, potassium, ATPase, and those locations are not always close enough to mitochondria, and therefore my guess is that neuron repolarization requires glucose, and that's the driver of this design principle that the brain runs on glucose. But it's a guess.
Concerning the entering of the glucose inside the neurons, there is a specific biochemical mechanism. Glucokinase is not controlled by insulin in the brain, the other tissue is controlled by insulin. So probably the fact that the brain is directly glucose could be depending on the direct effect of the glucokinase that infuse insulin.
Yeah, I mean, it certainly depends on the location of glucose transporters and glucokinase, and so I think what you say, you know, makes perfect sense. And so there's always a question whenever you look at these biological systems: what's the biochemical mechanism, what's the implementation, basically, and then what's the design principle, what's the purpose? And so that's a nice, likely explanation for the mechanism.
Glucose, as you see in your experiments, is kept at the same concentration in different conditions, homeostatic.
Mm-hmm.
Is there anything else here that's also kept at the same concentration, or are all the other things supply and demand driven?
I think everything is kept homeostatic. We're trying to prove that glucose is relatively unique in being kept so tightly homeostatic. And so we have a dream that most of the rest of the homeostasis is just mass action, and that glucose is very special in having this active homeostatic mechanism.
Quick question. The first one is, any idea about the source of the lactate?
As of today, my best guess is working muscle. So even when you're resting, there are muscles that are working. Breathing, for example. But we're trying to figure it out, and we don't have a great answer. We know it's distributed throughout the body, and so it has to be something distributed. Muscle is kind of everywhere on the body, but we're trying to figure it out.
The other one is, you used that analogy early on about two guys in a different color.
Yeah.
I mean, is there a way you can drip in glucose and lactate with different isotopes and then look at like
Yeah.
sense of
Yeah, you can put in one with the deuterium label and one with carbon-13 label, and amazingly, even though those have the same nominal mass, modern mass spectrometers can tell them apart. That's a really cool example of E equals mc squared, by the way. So that's the nuclear binding energies with the same number of protons, neutrons, and electrons. You get energy differences you can measure on a modern mass spec based on nuclear strong force. Anyway.
have you done that? Lactate and glucose differently and looking at particular tissues?
Short answer is no. We can talk. You can say more now, or we can talk about why you want to do it later.
Why do you say nuclear?
What?
Why do you say nuclear strong force with this?
Because it's the proton
Interaction between protons?
interaction between protons and neutrons in the nucleus is.
Integrated with pi zeros. It's not exactly.
Okay. That's good. You educate me later.
It has to be sure.
That's good. I want to be physically accurate on that as much as possible. Okay. Thank you, guys.